So there's the line of data there, 7b68, line 22, 7b68, flexible metal Ligands Apo-Holo pairs, 5G Wow Seti, alien radio signal is there, and I started working on this May 8, 2013. Today is June 24th. Flexible metals PDF results, Ligands Apo-Holo pairs, holo pairs, and cross-reference DNA molecule structure ligand binding when you get it to line 27 within line 17 formula ideas. SAM14 is a pulse sequence to PISEMA. It's continuing from several other videos, but the last one was called Immunogenic Polymer Bendable Metals UFO 5G Wow Seti. May 8, 2013, quote from a blog. From Biomol NMR, 2005 of June, can PISEMA experiments be used to extract structural parameters from mobile beta barrels? There's our list of scientists from Vancouver, BC, Canada. The link is www.ncbi.nlm.nih.gov. Keyword in the data is NalP autotransporter. We found microbiology on a quote from a blog of February 2013. Lipidation of the autotransporter NalP of Neisseria meningitis is required for its function in the release of cell surface exposed proteins. Keywords from the data idea was immunogenic protein metals from Biomed Science, March 1990. Polymer metal complexes of protein antigens, new highly effective immunogens. There's the scientist there from Russia, USSR Moscow. Keyword ideas from the data, immunogenic proteins, flexible metals. The proteins of May 2005, flexibility of metal binding sites and proteins on a database scale. Here's our list of scientists, again from Israel. Abstract protein metal binding sites in the pre-bound apostate and their rearrangements upon metal binding were not analyzed previously at a database scale. Such a study may provide valuable information for metal binding site prediction and design. A high-resolution, non-redundant dataset of 210 metal binding sites was created, containing all available representatives of Apo-Holo pairs for the most populated metals in the PDB. More than 40% of the sites underwent rearrangements upon metal binding. In 30 cases, rearrangements involved the backbone. The tendency for side chain rearrangement inversely correlates with the number of first shell residues. Analysis of side chain reorientations as a result of metal binding showed that in 95% of the rigid backbone binding sites, at most one side chain moved. Thus, in general, part of the first coordination shell is already in place in the pre-bound form. The frequencies of side chain reorientation directly correlated with metal ligand flexibility and solvent accessibility in the apostate. There's the link, www.ncbi.nlm.nih.gov. The keyword is Apo-Holo pairs. Sorry for the typo. Okay. On the diagrams we have A, B, C, D. Now this is a DNA ribbon that came up in the very beginning of the signal in line 17. If you look at those videos with the alien message, it shows these ribbons in it with the actual binary code. When we connect the ones and the zeros together with different colors, it actually looks a little bit like this. So my thoughts, this looks like the DNA alien formula that came up in line 17's data. It's like this too. Cross-reference DNA molecule structure when you get to line 27, the global structures of Apo and Holo proteins. The link is cssb.biology.gatec.edu. And here's again another set of shapes here. Data sets Ligand, free Ligand bound protein pairs, Apo-Holo.dat. Different Ligand bound structures, Holo.dat. And this is cssb.biology.gatec.edu. Okay, June 11, 2002, my thoughts. The keyword from the diagram is ligand binding, which comes up in the previous WOW videos too, adding this to the cross-reference. This Apo-Holo protein structure picture above is like a UFO spaceship. See how it's oblong like that? Okay, rotating the outside shell. So you have a shell inside, and like that's your ribbon of power, and then you've got this rotating on the outside of it. So the keyword SAM14 is a pulse sequence to PISEMA. Will be in our next video. Thank you for watching.